Welcome back to MLT 240. In this section of lecture, we're going to be talking about virology, and this section is going to be divided up into two parts. Let's first go over some terminology or some terms that relate to virology. The capsid is a protein that covers the virus. The nucleocapsid is the genome or genetic material of a virus that is enclosed in a protein coat. It's also referred to as just the capsid. The capsomere is one of the individual subunits that make up the capsid. The virion is the complete virus particle. The envelope is when the virus is surrounded by a phospholipid membrane. Syncytia is the giant multinucleated cell or cells formed from cell fusion as a result of virus infections. Latent, meaning there's no manifest or the virus is dormant or concealed, but it has potential or having the potential. Genome is the complete complement of genetic material on a cell or carried by an individual. Let's go over some general characteristics of viruses. Viruses are considered to be non-living. Viruses are not cells. They do not have cell membranes and other components of living cells. Viruses are obligate intercellular parasites that cannot multiply by binary fission. They must have living host cells for their reproduction. Outside of the host, they act as non-living chemicals. Also, viruses do not metabolize or have response to stimuli. They have genetic material and can therefore mutate and evolve. They do form parasitic relationships with living organisms. The viruses benefit at the expense of the living organisms. They are very small in size. The size range for most human viruses is in the range of 20 to 250 microns. So let's talk about the structure of viruses. Viruses lack mRNA or messenger RNA, and messenger RNA is involved in protein synthesis, but viruses do contain DNA or RNA. Viral DNA and RNA is transcribed into messenger RNA through the use of the host cell ribosomes. And in the ribosomes, they undergo the process of combining amino acids to produce proteins. Viruses may be specific for plant, animals, or humans, meaning they're only going to affect certain species. Viruses are going to come in a variety of different sizes and different shapes. And as we mentioned on the previous slide, they tend to be very small. And we actually could not see viruses until the early 1900s with the invention of the electron microscope. The virus particle or the virion has the capsid protein coat composed of repeating subunits that encloses the genetic material of either DNA or RNA. This coating protects that genetic material, and it also enables attachment and penetration of the host cell. The capsomere or the capsid is each subunit of the protein. The genetic material inside can be either single or double-stranded, so the structure can be linear or circular. The nucleocapsid is a combo of genetic material and the capsid. The envelope, of course, is the outer membrane. It's composed of a phospholipid layer with glycoproteins and matrix proteins, and the matrix proteins helps connect the capsid to the envelope. 6 stages of viral replication. The first is attachment. The virions must absorb or attach to a cell surface. Remember, you have to have a living organism for the virus to be active. You then have penetration, you have fusion or phagocytosis, and this is when you have the injection of the viral material. Then there's the uncoating process, which is the loss of the capsid. Then number 4 is replication and expression of genetic material. And in this step, there's a template of production for messenger RNA that happens, and it codes and directs synthesis of proteins in host cytoplasm and capsid in cytoplasm or the nucleus. Number 5 is assembly. The genetic material assembled into the protein coat. And number 6 is the release or lysis or budding, and may spread to local tissues or through blood. So this is where the process of the virus actually starts to spread beyond the cell that's being affected. Here is a diagram that'll hopefully help explain those steps that we just looked at. So here's the virus coming in contact with a human body cell. So the virus first has to attach to this body cell. Once the virus enters the cell, and you can see the process of phagocytosis is happening, the substances in the cell begin to strip off the virus's outer coating of the protein. Then the nucleic acid in the center of the virus is released. So here's the nucleic acid, that genetic information. The nucleic acid gets into the cell chemical manufacturing system. So it's basically the virus is taking over the cell. The cell ignores its own chemical needs and switches to making new virus. So the virus is starting to replicate within the cell. And the cell is sometimes destroyed by this process, and many of the newer viruses are released to other cells so they can affect those other cells. And sometimes these viruses will go to the blood so it can spread throughout the body. So what are the modes of transmission for viruses, or how do we become infected with a virus? Of course, there's respiratory aerosols that we can breathe in. We can become infected with a virus through contaminated food or water, the genital route, contaminated body fluids such as blood, urine, semen, mucus, through animal bites or insect bites, we can become infected with a virus, through endogenous latent infections. So these are variables that have laid dormant. Maybe when you become immunocompromised, they show up. An example might be shingles from the varicella zoster virus. Now let's talk about viral specimen selection, collection, and processing. The samples should be collected from the site directly during early stages if possible, and you want to collect during viral shedding, so when the virus is being released into the environment. So this is when someone is actually coughing, sneezing, things like that, when the virus could be being spread. Specimens usually are collected on swabs. Must be either rayon or dacron swabs. Calcium alginate swabs inhibit the replication of some viruses, so it can interfere with nucleic acid amplification tests. There's also a viral transport media, or VTM media, that can be used. It's usually a buffered saline and a protein stabilizer. And sometimes it also has antibiotics and antifungals to inhibit bacterial and fungal growth. These are usually used for respiratory, swab, and tissue samples. You want to collect specimens such as blood, bone marrow, amniotic fluid, urines, pericardial fluid, pleural fluid. All should be collected on the viral transport media. Of course, as I mentioned before, the viral transport media contains antimicrobial agents such as penicillin, gentamicin, and amphotericin B, and of course it's used to inhibit bacterial and fungal growth. For viruses, if you're collecting tissue samples must be kept moist. Do not place on media unless specifically designed for viral preservation. So you can use the viral transport media, you can use saline, or you can use tryptocase soy broth that can be added to sterile containers to keep the tissues from drying out. As far as processing tissues, you want to process promptly to increase detection of the virus, otherwise store at 4 degrees Celsius. Tissue samples can be frozen if processing is delayed greater than 4 days, but they need to be frozen at deep freeze at -70 degrees Celsius. Don't freeze at just -20 because ice crystals can form in the tissues. Laboratory methods for diagnostic virology. The clinical lab usually uses 4 major methods to diagnose viral infections. The first is direct detection of the virus in clinical specimens. Then there's nucleic acid-based detection, so you're going to be testing for the RNA or DNA. Then number 3 is isolation of viruses in cell cultures. So you're actually using cells that we have grown in the lab to see if they are affected by a virus that you're looking for. Number 4 is serologic assays to detect antibodies to a virus. So let's talk about each one of these 4 diagnostic methods a little bit more in detail. Let's first talk about direct detection. In general, this method is not as sensitive as culture, but it's faster. It goes ahead and allows for therapy of patients or treatment of patients. You can use a bright field microscope or an electron microscope with this particular method. You can go ahead and look for cytopathic effects or CPE. This is basically looking for structural changes in host cells. You can use direct fluorescent antibody tests or DFAs. So you're basically using a fluorescently labeled antibody to detect a certain antigen with direct fluorescent antibody testing. You can use enzyme immunoassays or EIA. This is the most popular viral testing method for hospital-based labs. So in this particular test, the virus is detected by colorimetric or optical density changes. So some type of color reagent is added and you're looking for a color change if the sample is positive for a virus. This is less sensitive, again, than cell cultures or immunofluorescence. For nucleic acid testing or NAT, this is a molecular testing method that looks for the presence of a specific genetic material. So you're looking for viral DNA or RNA that would identify the presence of the infection with a certain organism or disease. So you're looking for the RNA or DNA of a certain virus. So there's certain advantages versus disadvantages to nucleic acid testing. So let's go over the advantages first. First off, it's much faster turnaround time for this particular type of testing. There's better sensitivity compared to that of cell culture and DFA testing. The assays are quantitative. You can detect viruses that are non-culturable by cell culture. You can have the ability to detect multiple viruses simultaneously. You can run multiplex tests. And the potential characterization of a virus genetically. So you can basically genotype a test using nucleic acid testing. Here are the disadvantages of nucleic acid testing. That you can detect active and inactive viruses if there's RNA and DNA still available. The cost is very high of these tests. The need for specialized training and more complex facilities are there for these tests because these are highly complex tests. The lack of assays approved by the FDA. So things have come around a little bit more since the whole COVID pandemic, but there's still a lot of things that need to be approved and assays that need to be approved for this type of testing. And these type of systems will help epidemiologists, infectious disease physicians, and other public help by rapidly identifying viral pathogens during an outbreak. Some different examples of nucleic acid testing. There's hybridization assay. An example is FISH. So you're looking for a specific section of genetic material. There's the traditional polymerase chain reaction, or PCR, and real-time PCR. or RT-PCR, and we'll talk a little bit more about PCR here in just a second. Then there's the branched DNA assay, which is a signal amplification test as opposed to target amplification. It's used to detect nucleic acid molecules. And then there's the nucleic acid sequence, which is based on amplification. And then there's the combination of PCR and flow cytometry. In flow cytometry, you're basically detecting a certain type of cell. For PCR, or polymerase chain reaction, it's becoming more sensitive and more widely used for viral identification. And in PCR, you're going to amplify or make many copies of a selected region of DNA or RNA through several cycles of denaturing and polymerization with synthetic primers, resulting in more DNA copies, which can be identified with probes of known labeled sequences of DNA that are used to detect complementary sequences of target nucleotides. So you're basically making lots and lots of copies so you can identify what is there genetically. Make sure to take time to watch the link for the PCR video that is posted in Moodle so you can see exactly how PCR works. Viral isolation is still considered the gold standard method for virology diagnostics. There's three methods used for isolation of viruses. There's cell culture, and it's most commonly used, and it's done on living tissue. There's also animal inoculation, and this is extremely costly, and it's only used in special cases or in research or reference laboratories. And there's embryonated eggs that can also be inoculated, but this method is rarely used. For serological assays, it detects circulating antibodies to viruses after exposure. There are some inherent problems with this methodology. It measures the host response rather than directly detecting the virus. Antibody producing capabilities of human hosts differ widely. Antibody levels do not necessarily correlate with the acuteness or activity level of the infection because this is also host dependent. There's a fourfold rise in the titer in serum samples required to establish a diagnosis of a recent infection. With a few exceptions, some assays are able to distinguish between IgM, the acute infection, and the IgG, a previous infection. For serologic assays, the indications for testing by this method, you can do diagnosis of infections with non-culturable agents such as hepatitis viruses. You can do diagnosis of past IgG or acute IgM infections with various viral pathogens. The determination of immune status in regards to rubella, measles, VZV, hepatitis A or hepatitis B viruses. You can do monitoring of immunosuppressed or transplant patients. Serologic assays also allows for epidemiologic or prevalence studies. Now let's talk about the classification of viruses. They can be classified as either DNA viruses or RNA viruses, but never as both. The viruses in this unit are going to be discussed in groups based on their nucleic acid type. So we can have double-stranded DNA. It's going to be referred to as dsDNA, single-stranded DNA or ssDNA, or double-stranded RNA, dsRNA, or single-stranded RNA, ssRNA. The hepatitis viruses discussed are one group because they do not have the same type of nucleic acid. Okay, so this is going to conclude part 1 of virology and viruses. In the second part, we're going to be talking about some different examples of viruses and whether they are RNA, DNA, and whether they're double-stranded or single-stranded.